So good afternoon, everyone. I'm happy today to have the opportunity to present our work from the Neurocentre Magendie in Bordeaux. So today I will talk about the therapeutic administration of the borna virus X protein in SOD1 G93A mouse. So first, the context of the study is that among the hallmark of ALS, we have seen that there are mitochondrial alterations. These alterations are morphological. We've illustrated their swollen and vacuolated mitochondria, but also functional. Indeed, in the study conducted in our lab, we have shown that there is a decrease in the total and mitochondrial amount of ATP in mutated third culture motor neurons. And this agrees with the computational model of Professor Masson, according to which a decrease in ATP may induce an increase of motor neuron depolarization, which may finally lead to motor neuron degeneration. Therefore, mitochondrial impairment play a key role in the process of degeneration. And thus, a therapeutic track for ALS is to restore mitochondrial function. In this way, we are actually interested in the X protein of the borna disease virus. This borna disease virus is a non-cytolytic virus, and this non-cytolytic property is actually due to the X protein. This X protein has several localizations, nuclear, cytoplasmic, but interestingly, this X protein is also expressed in the mitochondria. And even more interesting, by this mitochondrial localization, not the other one, this X protein inhibits apoptosis in vitro, but also reduces neuronal loss in a model of Parkinson's disease. Actually, these very interesting properties are due to the last 29 amino acids of the protein, which constitute the PIX3 peptide. Based on these knowledges, in the previous study, we have co-administrated both PIX3 peptide via intranasal instillation and the X gene cloning vectors via intramuscular injection to SOD1 G93A mice. And from this, we have shown that we reduce motor neuron loss, we delay motor impairments. However, we haven't got any effect on the lifespan. That's why the aim of the project I will present the time I have left was to potentiate the therapeutic effect of the X protein. Well, how to do that? Since this therapeutic effect of the X protein are due to its mitochondrial localizations, and since this X protein only partly target mitochondria, researchers have modified the inosure X protein, replacing an aspartate in 4,3-U by an alanine, and by doing this, they obtain what they call the XA4 protein, which has an increased localization in the mitochondria. And due to this increased localization in the mitochondria, this X protein have increased neuroprotective effect in vitro. So the point was to use this XA4 protein. And the second point was to improve our administration way using AAV10 vectors, since AAV10 vectors are known to have effective transduction level in the spinal cord. So here is how we proceed. First, our collaborators in Paris have cloned either the gene encoded the X protein or the gene encoded the XA4 protein in AAV10 vectors. Then, at birth, we administrated via intracerebrovascular injection either AAV10X or AAV10XA4 to the mice. The first point of the study was to assess the effect of this approach on the motor performances of the animals. So, from the age of 60 days, the animals underwent several motor tests, grip tests, inverted grid tests, and rotarod. And here are the results for this first part. In white, we have the unaffected animals' grip strength, and as you can see there, they are quite normal. And in blue, you can observe that for untreated affected animals, we have a decrease in the grip strength as the disease progresses. And in green are the animals that receive the X protein. And as you can observe that for forelimbs, we actually delay the motor impairment, and for hindlimbs, we tend to delay this motor impairment up to 115 days there. And when we look at the inverted grid scroll of the animals, actually, we also delay the motor impairments with the X treatment. With the rotarod, we have a pretty similar profile. since also with the X treatment, we have a delay of the motor impairment. So what we can conclude there is that X treatment improves motor performances of the animals. What about the XA4 treatment? Actually, with this XA4 treatment, as you can observe there, we have very surprising results since for forelimbs and hindlimbs grip test, this XA4 treatment has no effect on the grip strength. However, it's very surprising since this XA4 seems to also improve motor performances for inverted grip test and rotor. The second part of the study was to assess the expression of either X or XA4 at the level of the motor neurons and also the motor neuron survival. So to do so, after the injection, a group of mice was euthanized at the age of 90 days. And we removed the spinal cord and we realized some immunohistochemistry to label motor neurons with SMI32 antibody and X expressing cells with an anti-X antibody. So here are the images obtained from this. In green, we have motor neurons, and in red, we have X expressing cells. And when we merge both images, you can observe that actually all the motor neurons express the X or XA4 protein. And this marker is pretty specific since the saline animals has no X-positive cells. When we quantify the number of SMI32 positive cells, here you can observe that we have already a decrease of the number of motor neurons at this time point of P90 for the untreated control animals. When we had our treatment either with X or XA4, we can observe that actually we improve the motor neuron survival. So both X and XA4 treatment allow to increase the number of motor neuron survival at this time point of P90. The last point of the study was to assess the effect of our approach on the lifespan. So here are the results. As you can observe there, we have the percentage of animals alive as the degrees progress and the average lifespan on the right. And again, despite our approach with AAV10 vectors, we don't have any effect on the life expectancy with the X protein. What about the XA4 treated animals? Well, these XA4 treated animals represented in red have actually an improvement of their life expectancy. So this XA4 treatment increased the life expectancy of mice. It's a pretty encouraging result since we have used a large cohort of animals and we have done this 3 times and we obtained the same result. So to sum up, in this study we have used 2 homologous proteins, X, which partly target mitochondria, and XA4, which has an increased localization in mitochondria. And both proteins have been in a vector, AAV10. And we put this on, injected this to SOD1 mice. And from this, we have shown that both X and XA4 protein improve lumbar motor neuron survival at P90. However, only with X protein, we improve motor performances, while only with XA4 protein, we improve the life expectancy. And the question from this study is why? Why do X and XA4 protein have so different effects? So it's the question for the next step of the study, and we already have some tracks to answer. And the point was to answer the first difference at the level of a motor, at the level of a life expectancy. So the point will to look at the phrenic motor neurons, since these motor neurons control respiratory muscle. So maybe with X protein, we have less preservation of these phrenic motor neurons, while with XA4, we better preserve these phrenic motor neurons. The second point is the difference at the level of motor performances. So in this point, we will look at the effect of our approach on the neuromuscular junction preservation. So maybe with the X protein, we have better preservation of the neuromuscular junction, while with XA4, we have less or no preservation of a neuromuscular junction. Well, this brings me at the end of my presentation. I would like to thank all my team at the Neurocentre Magendie in Bordeaux, and particularly Stéphanie Chevalier, my supervisor, and our group leader, Grendal Masson. I would like also to thank our collaborators in Paris for the vectors, but also the FRM and ASLA for their financial support. ASLA financed my PhD, so thank you for that. And finally, I would like also to thank the Festland organization, since they trust us and financed a 4th year of PhD, which we hope will help us to answer the question I just raised. And thank you for your attention.
Thank you very much. Any question in the crowd? No?
Thank you. Hi, very nice presentation. I just wonder if you looked at the activity of motor neurons. Does this treatment change the properties of motor neurons, their excitability, or anything similar?
Actually, we don't look at these properties. We have just seen that this X protein is expressed in a large part of the brain and the spinal cord, but not the specificity of which kind of neurons or which activity of these neurons.
No question?
Nice presentation, because you're targeting mitochondria. So I was just wondering if you look at the mitochondria, in the presence of the virus treatment.
Yes, it's a good point. Since this project is ongoing, it's another part of the project to do that.